December 18, 1944, Hürtgen Forest, Germany. Oberarzt Heinrich Müller unsealed the seized American medical kit. Within, amidst the usual bandages and morphine syrettes, he discovered little glass vials marked with a term he did not recognize. In the next 72 hours, this German army physician would observe an event that defied all his knowledge from 15 years of military medicine. American servicemen with infected wounds that ought to have been fatal were ambulating, speaking, and going back to the fight. His own patients, suffering the same injuries, were perishing at a tenfold pace. By December 1944, the Wehrmacht Medical Corps confronted a predicament that no degree of tactical skill could overcome. Mortality rates from wound infections had stayed almost static since World War I. Roughly 30% of soldiers with penetrating injuries acquired lethal infections, despite prompt care. The figures were harsh and steady on all fronts. German Army medical data, 1944, abdominal injuries with peritonitis, 90% fatality. Compound fractures with gas gangrene, 75% fatality. Chest injuries with empyema, 60% fatality. Basic infected wounds, 30% fatality. Oberarzt Heinrich Müller had watched these statistics become individuals. 19-year-old servicemen who endured the trauma, just to perish 5 days later from infections that made their blood septic. The German medical strategy depended on three main techniques. 1. Sulfonamide drugs, brought in during 1935, somewhat effective against certain bacterial infections, but with a narrow spectrum and considerable adverse effects. 2. Surgical debridement, the vigorous extraction of infected tissue, frequently necessitating amputation to stop the spread of sepsis. 3. Antiseptic fluids, topical use of iodine, mercurochrome, and carbolic acid that destroyed bacteria but also harmed healthy tissue. These techniques were the peak of 1944 military medical science. They were also basically insufficient. Heinrich Müller finished his studies at Heidelberg University Medical School in 1929. He had been a Wehrmacht medical officer from 1939, cycling through Poland, France, Russia, and currently the Western Front. By December 1944, Müller was in charge of a battalion aid station 800 meters back from the front lines in Hürtgen Forest. His facility comprised one surgical tent with 2 operating tables, 40 cots in modified bunkers, 6 medical assistants, diminishing stores of sulfonamide powder, and no antibiotics besides sulfonamide derivatives. His daily work had turned bleakly routine. Sort arriving casualties, stabilize them for removal, and observe 30% acquire infections that descended into sepsis regardless of all efforts. The Hürtgen Forest campaign exacerbated it all. The close-quarters forest combat resulted in terrible wounds polluted with dirt, wood splinters, and cloth forced deep into tissue by fast-moving shrapnel. On December 16th, American troops initiated a counter-offensive. Müller's aid station was momentarily overtaken, then reclaimed by German units 6 hours afterward. The Americans had withdrawn rapidly, abandoning gear they were unable to transport, which included medical equipment. December 18, 1944, 0600 hours, Müller took stock of the seized American medical provisions. The majority of items were recognizable: morphine syrettes, plasma bottles, bandages, sulfur powder. Then he discovered the little cardboard containers marked Penicillin, 100,000 units for military use only. Every box held 5 glass vials of a yellowish powder. The directions were written in English. Yet Müller's medical education involved sufficient English to grasp the essentials. Reconstitute with sterile water, administer intramuscularly every 3 hours, maintain treatment for at least 5 days. The dosing appeared ridiculously frequent. Every 3 hours required rousing patients all night long for injections. The treatment length, a 5-day minimum, would use up massive amounts of the medication. Müller had picked up whispers regarding penicillin. German intelligence briefs noted it as early as 1943, detailing it as a novel antibiotic material being developed in Allied labs. He, however, had never viewed it, never employed it, and possessed no working knowledge of its impact. He confronted a choice. He had 23 patients at present under his supervision. 14 possessed infected wounds. 5 were worsening towards sepsis, notwithstanding sulfonamide care. The seized penicillin stock was finite, possibly sufficient for 3 or 4 patients if he adhered to the American dosing regimen. Standard Wehrmacht medical policy forbade using unknown medications on German soldiers. The danger of negative reactions, allergic responses, or unrecognized side effects was deemed intolerable. But Muller had observed 47 men perish from wound infections in the past 6 weeks. The conventional treatments were failing. He chose 3 patients for the test. Patient 1, Gefreiter Klaus Weber, age 21, compound femur fracture, December 14th. Wound infection progressing despite sulfur application. Fever 39.2 degrees Celsius, wound discharge purulent. Outlook 60% chance of death within 72 hours. Patient 2, Unteroffizier Ernst Hartmann, age 27, shrapnel injury to left shoulder, December 15th, gas gangrene suspected in deep tissue. Fever 40.1 degrees Celsius, crepitus felt around the injury. Outlook 75% chance of death, amputation probable. Patient 3, Obergefreiter Franz Schmidt, age 24, abdominal injury with suspected peritonitis, December 16th, stiff abdomen, rebound tenderness present. Fever 39.8 degrees Celsius, blood pressure falling. Outlook 90% chance of death within 48 hours. Muller gave the first penicillin injections at 0800 hours, December 18th. He adhered to the American protocol precisely. 100,000 units intramuscularly, replicated every 3 hours. The initial 24 hours revealed no drastic shifts. Fever stayed high. Wound discharge persisted. Muller recorded everything in his medical journal, anticipating the experiment's failure. December 19th, 600 hours, 22 hours post-initial injection. Weber's fever had fallen to 38.1 degrees Celsius. Wound discharge had lessened by roughly 40%. The patient noted diminished pain. Hartmann's shoulder injury exhibited reduced swelling. Crepitus was not as distinct. Fever lowered to 39.3 degrees Celsius. Schmidt stayed in critical condition, but his blood pressure had steadied. Abdominal stiffness slightly lessened. December 20th, 600 hours, 46 hours post-initial injection. Weber's fever returned to normal at 37.4 degrees Celsius. Wound discharge was minimal, tissue granulation apparent. Patient was upright, consuming solid food. Hartmann's gas gangrene advancement had ceased. Injury margins revealed healthy tissue. Fever, 37.9 degrees Celsius. Amputation was not required anymore. Schmidt's peritonitis indications were resolving. Abdomen was soft, bowel sounds were coming back. Fever 38.2 degrees Celsius. Patient was aware and responsive. Muller had performed military medicine for 5 years on 4 fronts. He had never observed an infection reversal so fast, so total, so uniform among several patients. By December 22nd, all 3 patients given penicillin were recuperating. Weber was ambulating with crutches. Hartmann's shoulder injury was healing. Schmidt was consuming food and asking to return to service. Concurrently, 2 other patients with comparable infections, managed with standard sulfonamide regimens, had perished. Muller started to compute the consequences. Standard Wehrmacht infection care, 1944, sulfonamide medications, 40 to 50% effective against vulnerable bacteria. Surgical debridement needed in 60% of instances. Amputation percentage for limb infections, 35%. Total mortality for infected wounds, 30%. Penicillin care, noted outcomes, 100% efficacy against observed bacterial infections. Surgical debridement unneeded in 2 of 3 instances. Amputation averted in 1 of 1 prospects. Fatality, 0 of 3 patients. The statistical fact was astounding. If these outcomes were typical, penicillin lowered infection fatality by a 10-fold factor. Muller sought data from Wehrmacht Medical Command regarding German penicillin manufacturing. The reply arrived December 28th. German penicillin manufacturing, 1,944, 8 to 10 kilograms annually. Estimated American manufacturing, 1944, unclear but presumed to be substantially greater. The real figures, which Muller would not learn until after the war, were more staggering. Allied penicillin manufacturing, 1944. United States, 1,633,000,000 units per month by June 1944, sufficient to manage every Allied casualty needing antibiotics. Output rising tenfold every 6 months during 1943 to 1944. German penicillin manufacturing, 1,944, roughly 8 to 10 kilograms total yearly output. Enough to care for under 1,000 patients. No industrial-scale manufacturing capability. Research was split among multiple rival institutions. The technological disparity wasn't solely about finding penicillin. British scientist Alexander Fleming had pinpointed penicillin's antibacterial qualities in 1928. German scientists were aware of it. The disparity was industrial manufacturing ability. The United States had converted penicillin from a lab oddity into a mass-produced military medical item using deep tank fermentation methods. Large-scale bioreactors generating thousands of liters, corn-steep liquor substrate, an agricultural byproduct that boosted penicillin harvest tenfold. Strain improvement. Mutated penicillium strains yielding 1,000 times more penicillin than Fleming's initial samples. A unified government-industry collaboration. 21 firms manufacturing penicillin under common protocols. Germany possessed none of this framework. German penicillin research stayed in minor university labs making gram amounts while American plants made tons. The penicillin manufacturing gap created a survival rate difference that became statistically quantifiable by late 1944. Allied casualty infection fatality, 1,944. Wound infection deaths, 3%. Down from 12 to 15% in WWII. Pneumonia fatalities under 1%. Down from 18% in WI. Amputation percentage for infected wounds, 3%. Down from 8% with conventional care. German casualty infection fatality, 1944. Wound infection fatalities, 12 to 15%, static from WWI levels. Pneumonia fatalities, 18%, no betterment from WI. Amputation percentage for infected wounds, 8%, no betterment from conventional care. The figures exposed an unseen technological tool more destructive than any tank or airplane. By December 1944, American units had access to 4 million sterile penicillin packets per month, sufficient to manage every casualty needing antibiotics. German units had enough penicillin for topical wound dusting only. Injectable penicillin failed to reach German field hospitals until October 1944, and then in amounts adequate for fewer than 1,000 patients overall. Muller recorded the disparity in his medical journal. American prisoners of war with the same wounds as his German patients recuperated at rates 10 times greater. The variation wasn't medical expertise, it was pharmaceutical industrial capability. The penicillin disparity represented something bigger than military medicine. It was industrial manufacturing principles applied to human existence. American penicillin production strength, 1944, deep tank fermentation methods. Large-scale bioreactors generating thousands of liters. Corn-steep liquor substrate. Agricultural byproduct boosting harvest tenfold. Strain improvement. Mutated Penicillium chrysogenum yielding 1,000 times more than Fleming's initial samples. Government-industry collaboration. 21 firms manufacturing under common protocols. German manufacturing constraints. Fragmented research, numerous rival university labs, minor-scale manufacturing. Gram amounts versus American tons, resource restrictions. Raw materials rerouted to weapons manufacturing. No industrial unification, every facility running independently. The outcome was a medical technology disparity that translated directly into combat readiness. American divisions kept 90% strength after major battles owing to swift infection recuperation. German divisions dropped to 60% strength as infections took soldiers for weeks or permanently. Muller understood he was observing the Industrial Revolution's last stage. Not merely mass manufacturing of armaments, but mass manufacturing of human existence itself. December 30th, 1944. Muller finished his last penicillin treatment journal. 3 patients recuperated completely. 2 additional patients with comparable infections, managed with conventional regimens, had perished. The seized American medical kit held sufficient penicillin for maybe 20 patients in all. Muller had employed it on 5. He recorded everything fastidiously, aware this data would be valuable to Wehrmacht Medical Command. But he also understood the wider truth. Even if Germany was able to manufacture penicillin in American amounts, the industrial framework didn't exist. The penicillin disparity was not solely about one medication. It was about the basic distinction between societies, set up for total war manufacturing versus those set up for gradual military requirements. America had converted penicillin from lab oddity to mass-produced military item using agricultural byproducts, unified industry, and government funding. Germany had kept penicillin in university labs, making gram amounts while American plants made tons. Muller endured the war and was taken prisoner by American units in May 1945. He was a POW for 3 years, during which time he had access to American medical journals detailing penicillin production statistics. The figures validated his 1944 observations. By the war's conclusion, American penicillin output had reached 646 billion units, sufficient to manage every Allied casualty needing antibiotics with excess. German manufacturing stayed at 8 to 10 kilograms yearly, enough for fewer than 2,000 patients. The unseen weapon that preserved more Allied lives than any tank, aircraft, or artillery unit wasn't a weapon at all. It was industrial medicine applied to human existence. This narrative is based on recorded historical trends of penicillin manufacturing and application during World War II, while Oberarzt Heinrich Muller is a composite figure symbolizing German medical officers who came across American penicillin. The manufacturing data, fatality rates, and technological disparities detailed are factually correct. The penicillin manufacturing difference between Allied and Axis forces signifies one of history's most important instances of industrial capability deciding military medical results. The next time you consider World War II's technological advancements, recall the invisible weapon that preserved more lives than any tank or airplane. Occasionally, the most potent weapons aren't created to annihilate. They're created to restore.